This lecture covers the urinary system. Part 1 is simply to learn the anatomy of the system. I'm pretty sure you've covered this in lab already, so I'm not going to spend a lot of time on it. You know that you have pairs of kidneys. Each kidney has a ureter attached to it. Empties into a urinary bladder, which has a urethra. Urination or micturition is the process of eliminating urine. We'll be doing that at the very end. 3 functions of the urinary system include excretion, which is the removal of wastes, elimination, which is the discharge of those wastes, and most important in my opinion, homeostatic regulation. The wastes that are removed so that you can maintain homeostasis. Homeostatic regulation in that are responsibility of the kidneys include blood pressure and blood volume, which we've already studied, so we're already somewhat familiar with things like renin, the enzyme secreted by the kidneys to stimulate production of angiotensin. We know that the hormones aldosterone and antidiuretic hormone have their effects at the kidneys, so we're finally going to see where those effects and how those effects are going to work. Kidneys are very important in regulating ion concentration, so any kidney problem, kidney disease, can lead to a loss of homeostasis of these ions. Most important, sodium, potassium, and chloride, but also calcium, and calcium is regulated hormonally, as we've already seen. So homeostatic functions of the urinary system also include stabilizing blood pH. This is probably going to be emphasized more in the next lecture, which is going to cover acid-base balance. We'll also come back to a lot of these other homeostatic mechanisms of controlling blood volume and controlling blood electrolyte balance in the next chapter. So we'll be coming back to the kidneys again after we finish this unit. Conserving nutrients, this includes not just organic nutrients, but also things like calcium ions, which are reabsorbed by the kidneys, and reabsorption of organic things like glucose. And then this is probably an under-described function of the kidneys. We don't have any specific examples we're going to examine, but detoxification of poisons and excretion of metabolic poisons like, for example, fungal toxins are excreted by the kidneys. All right, so we're going to talk next about the functional anatomy of the kidneys. If there's anything on the actual gross anatomy of kidneys that you need to know for lab, I'm sure you've already covered it there, and I'm just assuming you know that. So we're going to focus in on the functional anatomy. The kidney is divided into really 3 parts, technically 4. The 3 parts that contain the structures of the kidney that actually do the functions of the kidney are the renal cortex, the renal pyramids, and the renal columns. Renal columns are just an extension of cortex. Cortex and columns are basically the same histologically. They contain a lot of the same structures. So cortex and columns versus pyramids. And when we get to the different parts of the nephron, we'll see where these things, where these structures are. The last part of the kidney, the 4th part, is the hollow interior of it, where urine is going to collect and get propelled through the ureters. A kidney lobe consists of a single pyramid and the surrounding cortex and columns. So once you've explained one lobe of the kidney, you've kind of explained them all. Ducts within each renal papilla are going to empty into a minor calyx, which the minor calyces join to form the major calyces, which empty into the renal pelvis. So that's our 4th part. You have renal column, renal pyramid, renal cortex, and then these hollow spaces, the calyces and the pelvis, which drains into the ureter. And of course, we've got a nice diagram of that, which I'm sure you've already looked at in lab, or you will look at in lab. The blood supply to the kidneys is really an important thing to figure out, because the main job of the kidneys in maintaining homeostasis and eliminating waste is that it's got to clean the blood. It filters the blood. So blood supply to the kidneys is way more than you would expect it to be if you just thought, well, the kidneys need a blood supply. They get about 25% of the total cardiac output at rest. So that's your 2 little fist-sized kidneys getting 25% of your total blood flow at rest. Now, when you're actively exercising, the total amount of blood flow increases. but the percentage received by the kidneys is going to decrease. About 1,200 milliliters a minute at rest, and maybe only 600 or 650 milliliters a minute during intense activity. The blood supplies through the renal artery. It subdivides into segmental arteries, which subdivide into interlobar arteries. Interlobar arteries run between the pyramids. At the top of each pyramid, they split into arcuate arteries. You can think of these as arching over the pyramids. And then within the cortex, each arcuate artery gives rise to little interlobular arteries, which give off the afferent arterioles. And the afferent arterioles supply individually the blood to the capillaries in the cortical material, that is in the renal cortex and the renal columns. The venous drainage is going to basically mirror the venous drainage mirrors the arteries. So if you have an interlobar artery, you have an interlobar vein. We'll see when we get into the capillary supply in the renal cortical material that at the capillary level it's a little bit more complicated, but here at the major arteries and veins they're very parallel, and each kidney is drained by a renal vein. So here you can see a diagram showing you the blood supply. Okay, so once we get up into the cortex, you can see that the arcuate arteries give off branches that your book calls cortical radiate arteries, other books call interlobular arteries, and each one of these is going to give off an afferent or a bunch of afferent arterioles, and each afferent arteriole gives rise to a capillary ball called a glomerulus. Glomerulus just means little bowl. And then after the blood is drained, it goes through the radiate veins to the arcuate vein, to the interlobar vein, to the segmental vein, to the renal vein. So it's just going to be very parallel. So you've got this nice diagram showing you that. Now within the nephron, down in the box at the bottom, from the glomerulus, which remember is a ball of capillaries, blood is drained into, instead of venules, arterioles, which is unusual, and we'll talk about that. The efferent arterioles drain into peritubular capillaries, and those will drain into venules. Okay, so a little bit on the renal nerves. Renal nerves are part sensory and part motor. The sensory function is mostly to react to stretching, so that as fluid accumulates in the kidneys, it's going to drain properly. And in addition to that, probably awareness of the blood pressure in the kidneys and perhaps the composition of fluid in the kidneys. The motor branch of the renal nerves is mostly sympathetic, and sympathetic innervation can do a couple of things. It can cause vasoconstriction of the arteries within the kidneys, including the afferent and efferent arterioles, as we'll see. And also stimulates the release of renin, which you remember helps regulate blood pressure. So the nephrons are the functional units of the kidneys. There's probably about 1 million nephrons in each kidney. Each one consists of a renal corpuscle and a renal tubule, and then the renal tubule is going to empty into the collecting system. The renal corpuscle just means little body. So the renal corpuscle is that glomerulus, that little ball of capillaries, and the tissue surrounding it, which is called the Bowman's capsule or glomerular capsule. And then the renal tubule is divided into regions, which we'll take a look at. So if you look at this diagram, we're not looking at the histology right now, just the big picture. So you can see an afferent arteriole leading into the glomerulus, an efferent arteriole leading out. The green arrow indicates the filtration of fluid into the capsular space. The outer wall of the glomerular capsule captures that fluid. We call it filtrate, and sends it into the first part of the renal tubule. Once it enters there, we call it tubular fluid. So we have a proximal convoluted tubule. Convoluted means folded or twisted. So you can see how highly convoluted it is. That leads into what looks sort of like a trombone slide. That's called the nephron loop. That leads up back to a distal convoluted tubule, and that's the end of the nephron. From there, it's going to go into the collecting system. So the glomerular capsule forms the outer wall of the renal corpuscle, and it captures the fluid that is filtered out of the capillaries of the glomerulus. The glomerulus is about 50 capillaries. That's a huge amount of capillaries. I've never seen a diagram that showed how complex it really is. Blood comes in through the afferent arteriole and leaves through the efferent arterioles, and that's going to impact the physiology of filtration as well. So that capsular outer layer is just simple squamous epithelium. It just holds that filtrate, that fluid, keeps it under some pressure, helps push it into the renal tubule. The visceral layer is really pretty complicated. The visceral layer completely covers all of the surface of all 50 glomerular capillaries, and we'll take a look at that. The capsular space is just where the fluid accumulates. The visceral layer is composed of cells called podocytes, and they're called this because they have little feet. They have foot processes called pedicels that wrap around the glomerular capillaries, and in between these, think of it as intertwining your fingers. There are spaces between them called filtration slits, and these filtration slits, 6 to 9 nanometers wide is not very wide, but the gap is large enough to let large amount of fluids leak through, but it pretty much stops things like plasma proteins from getting out. So looking at this figure, find the visceral layer labeled at the top right, and note that it is composed of these little foot cells, the podocytes, with their little feet called pedicels, and how they interact with each other, and how in pink underneath of them you can sort of see the capillary wall. Starting on the left at the bottom, you can see the afferent arteriole has circular layers of smooth muscle around it that let it vasoconstrict. You've got the juxtaglomerular complex. We'll talk about that later. And at the far right, draining out of the capsular space, is the proximal convoluted tubule. So glomerular capillaries are fenestrated capillaries. I hope you remember what that means. It means they have large fenestrae or pores in them. So fluid can really leak easily out of these fenestrated capillaries. That fluid is going to go through those filtration slits and into the capsular space. You actually produce about, I think it's about 180 liters of fluid a day, and you're going to reabsorb, mostly in the renal tubule, you're going to reabsorb about 179 liters of that 180 liters that filters through these capillary walls. There's another kind of cell here. It's the intraglomerular mesangial cell. For a long time, we didn't know what they do. Now it appears that in addition to supporting the glomerular cells, it also helps control the diameter of the capillaries. I don't test over it simply because it's just not entirely clear everything that the mesangial cells do. All right, so focusing in on just where the action is, the filtration membrane has 3 layers: the fenestrated endothelium, its basement membrane, and the foot processes of the podocytes. That's the membrane, and of course filtration goes through those fenestrae, the openings in the fenestrated capillaries, filters through the basement membrane, which is just like a layer of fabric, and then through the filtration slits that are produced by the podocytes. So in filtration, blood pressure forces water and small solutes into the capsular space. Larger solutes like plasma protein do not easily pass. A little bit might sneak through, but unless you have inflammation going on, you are not going to put a lot of protein from the plasma into that capsular fluid, so it is practically protein-free. All right, so here's a diagram showing you, let's start at the middle with the mesangial cell. It's just a big cell that kind of hooks the capillary walls to each other, somehow helps control their diameter, probably by pulling on them. I don't know exactly how that works. Most important here is the filtration membrane. So you can see the layers of the filtration membrane, and you just need to visualize fluid passing out of the plasma, through the basement membrane, through the filtration slits, and into the capsular space. All right, so moving on to the renal tubule, we've got the convoluted portions on each side, separated by the nephron loop, or the loop of Henle, in the middle. And we're going to see some individual variation here. Now we want to look at the histology. So look at the proximal convoluted tubule diagram at the top left. You'll notice these cells have microvilli at the apical surface, which is at the top of the picture, and then down at the basal end, they have lots of little folds. They're jammed and packed with mitochondria. On the far right at the top, the distal convoluted tubule, a little bit thinner wall, not hardly any microvilli, but still packed with mitochondria at the basal surface, which has a lot of folds. Those mitochondria are doing something down there. There are a lot of active transport processes that require ATP. In the middle, the renal corpuscle, as we already read, was squamous epithelium. And then in the nephron loop, we've got thin and thick portions. The thin portion is squamous. The thick portion is cuboidal cells, again, packed with mitochondria, so you know something's up. And then we also divide the nephron loop into descending limb and ascending limb. And there's no sharp border between the convoluted tubules and the loop. So it's more of a functional distinction. All right, so as fluid moves through the renal tubule, this tubular fluid, which started out as filtrate, is going to get acted on by the cells of the renal tubule. They're going to remove things that you want to keep, and in some cases they're going to actively secrete things you want to get rid of. The proximal convoluted tubule is that first segment. Remember, it has lots of microvilli, so it has a highly absorptive surface area. About 65% of the total fluid volume is going to get reabsorbed here. It says primary function is reabsorption of ions, but I'm going to tell you that almost all the organic nutrients are also reabsorbed here. There was glucose in what you filtered in that glomerular blood. There were amino acids. There were lactic acid, and these are all reabsorbed in the proximal convoluted tubule, so that by the time the fluid leaves the proximal tubule and goes to the loop of Henle, there are no organic solutes left. It is all water, salts, and wastes. So in the nephron loop, in the descending limb, fluid goes down towards the pelvis. In the ascending limb, it comes back up, and we have segments that are either thick or thin. And we're going to see salts are reabsorbed there, salts and water. In the distal convoluted tubule, we only have about maybe 15% of the total fluid volume left. The primary function here is to absorb water and ions, and also to actively secrete things that you're trying to get rid of. So, all right, now we're going to turn our attention to something that's not directly related to urine formation, but it is a regulatory area. This is the juxtaglomerular complex, and it consists of the macula densa and the juxtaglomerular cells, along with those mesangial cells. The macula densa cells are actually specialized cells of the distal convoluted tubule. They are receptor cells, so they can respond to changes in the osmotic pressure of the blood, or even just the total fluid pressure, not of the blood, of the filtrate, of the tubular fluid. The juxtaglomerular cells are smooth muscle cells in the wall of the afferent arteriole, and likewise they're functioning as baroreceptors, meaning they're responding to changes in the pressure of the blood, and these are the cells that secrete renin when your blood pressure is too low. Those mesangial cells provide some kind of feedback control. Remember, we're not entirely sure how they're doing what they're doing. So here's the same diagram again, but this time we're going to focus on the juxtaglomerular complex. So you can see that the specialized cells of the distal convoluted tubule form the macula densa, and then there's some mesangial cells in between that and the juxtaglomerular cells, which are modified cells of the afferent arteriole. All right, so as this fluid moves out of the renal tubule, that is, as it leaves the distal convoluted tubule, it moves into the collecting system. The collecting system is mostly collecting ducts. They start in the cortical region, but they run through the medulla, and to some extent they can modify the composition of the fluid in them and reabsorb water or not reabsorb water, depending on the state of hydration of the body. They end at the papilla, which is the tip of the renal pyramid, at what we call the papillary duct, which drains into the minor calyx. In humans, once that fluid leaves the collecting duct, there's no way to adjust urine composition. In other animals there is, but not in humans. So here you can see a collecting duct way up high in the cortex, running through the medulla, and then ending as a papillary duct. Cortical nephrons are those that have a pretty short nephron loop, and the nephron loop is mostly within the cortex. The juxtamedullary nephrons have an extra long nephron loop that goes down into the pyramids of the medulla. In humans, only about 15% of our nephrons have these extra long loops of Henle, and as we're going to see in the pictures, there's some differences in not just where they are and how long the loops are, but also in the blood supply. So in a cortical nephron, the loop of Henle is surrounded by peritubular capillaries, which also extend around the proximal and distal convoluted tubules. But in those different juxtamedullary nephrons, there are long, straight capillaries that run alongside of them called the vasa recta. And if you follow the vasa recta, you see that as they start down from the cortex, they go down around the nephron loop, they loop back up, and they empty into the vein, which is colored blue for your convenience. So they parallel the flow through the nephron loop. Fluid goes down and comes back up in the nephron loop. Blood goes down and comes back up in the vasa recta. And that whole structure then parallels the collecting duct, and we're going to see why that matters. All right, so that's a quick overview of the structures of the nephron and associated structures. As we get into renal physiology, things can get a little complicated. So stop, read, consider, read your book, make sure you understand this as you're going. So our goal in forming urine is to get rid of metabolic wastes, and you need to know what at least some of those metabolic wastes are. 3 that are really important to consider are urea, creatinine, and uric acid. All of these contain nitrogen, so we call these nitrogenous wastes. None of them are protein, so we call them NPN, non-protein nitrogenous wastes. Urea is the most abundant organic waste. Do you remember last chapter we were talking about deaminating amino acids so we could convert them to organic acids or interconvert them to something else? And that produced ammonia, which is really toxic. You can do that in the kidneys, and you can just dump that ammonia straight into the urine because it's not going to get in the bloodstream. But and that makes ammonia a 4th nitrogenous waste. But in the bloodstream, we really don't want a lot of ammonia, so all the amino groups that are deaminated in the liver are converted to urea in the liver. So urea is the most abundant of the organic wastes. Creatinine comes from breakdown of creatine in muscle cells. There's a certain amount of muscle cell turnover that's just a sort of a baseline normal, so there's always some creatinine. Note the difference between the words. Creatinine is the waste, creatine is the stuff in your muscle cells. And then our 3rd is uric acid. Uric acid comes from the nitrogen bases. Again, they just have a certain rate of breakdown. These are the adenine, guanine, cytosine, and thymine in your DNA and RNA, and uric acid is a waste product of DNA. Organic wastes, such as these three are organic wastes, are simply going to be dissolved in the bloodstream. They're going to get eliminated when the kidneys remove them, and they, because of osmotic reasons, are going to bring water with them. Okay, so kidneys usually produce a very concentrated urine to conserve water. The concentration of the urine can be as high as 1,200 milliosmoles per liter. Now, we haven't really used these units much, so to be able to compare that, the osmolarity of blood is about 300 milliosmoles per liter. And if you want to secrete a dilute urine because you drank a lot of water, the concentration can be as low as about 100 milliosmoles per liter. So that's quite a range from 100 milliosmoles per liter, very dilute, to 1,200 milliosmoles per liter, very concentrated, compared to blood osmolarity of about 300 milliosmoles per liter. So one of the things the kidneys do is they concentrate that filtrate, that is, they pull the water back out. And if you remember what I told you, you filter about 180 liters of water every 24 hours. you only have about 3 liters of plasma volume, so you're recirculating that fluid through your kidneys about 60 times a day. 60 times 3 would be about 180. So if you're not reabsorbing that water, then you're going to dehydrate fairly quickly. And this happens, it's not that common, but it can happen in certain disorders. Antidiuretic hormone, we know, is responsible for most of that water reabsorption. And we're going to see that people that don't produce enough antidiuretic hormone overproduce urine, and they have to just keep drinking water or they're going to dehydrate. And then there are valuable materials we want to reabsorb, not just sugars and amino acids, but things like calcium as well. All right, so as we get into the processes of urine formation, we're going to see there are 3 of these. Filtration occurs in the renal corpuscle. Reabsorption and secretion occur in the renal tubules. So we often emphasize that by calling them tubular reabsorption and tubular secretion. The tubular fluid is what is left of the filtrate. The peritubular fluid is the fluid surrounding the tubule, and it is in direct contact with the blood in those peritubular capillaries. So reabsorption is putting stuff back into the blood from the filtrate, and secretion is taking additional wastes out of the blood and putting them into the tubular fluid to get rid of them. So here you have a nice diagram showing you all the parts of the system. Please take the time to go over this. You can pause your video or you can study it in your textbook. Now, this is an important diagram because it's a table of values comparing plasma values to urine values, and you have to compare these to see which ones you're keeping and which ones you're getting rid of. So look at sodium, and you see in the plasma it's got a range of 135 to 145. The units here are milliequivalents per liter. And in the urine only, well, it goes from below that, 40, up to above that, 220. So it depends on how much salt that you're consuming. If you eat a lot of salt, you're going to excrete more of it. Potassium, look how low potassium level is in the blood. We'll come back to this next chapter. Only about 3.5 to 5 milliequivalents per liter. And in the urine, 25 to 100, which means you're almost always excreting potassium. Chloride, 100 to 108 in the plasma, 110 to 250 in the urine, which means, again, you're excreting some chloride. Bicarbonate level, very low in the urine, but it varies from 1 to 9. That's a huge range, and we'll see when we get into acid-base balance why that is. All right, metabolites. Glucose, 70 to 110 in plasma, almost none in the urine. Lipids, almost none in the urine. Amino acids, almost none in the urine. Proteins, none. These organic nutrients are not going to be lost in the kidneys. You're going to reabsorb all or almost all of them. And then look at the nitrogenous wastes. Here you see urea, creatinine, uric acid, and ammonia, and you see huge amounts of them in urine compared to plasma. That's your cue that these things are being gotten rid of. You are concentrating them in urine to excrete them. All right, so we're going to look at these processes in detail. Glomerular filtration is driven by hydrostatic pressure. That is the blood pressure. But it's not just simply blood pressure. It's the blood pressure being counteracted by competing pressures, as we'll see. So we have to get through that filtration membrane, and small particles can pass and large ones are not. And remember, that's our definition of filtration. Just like when coffee filters through a coffee filter, that the grounds, the large particles, do not end up in your coffee cup. Likewise here, the plasma proteins do not wind up in the filtrate. All right, so looking at this extreme close-up on the right, you can see the fenestrations or pores of the fenestrated capillary. You can see the basement membrane, which, remember, is just more like a fabric. It's not impermeable. It's just fibrous proteins with maybe a few glycoproteins, but it doesn't impede the flow of most things. And then the filtration slits, which allow just about anything through. So the glomerular capillaries are fenestrated capillaries. Those pores are small enough that blood cells can't go through. Plasma proteins can actually pass through here, but they're not going to get past the basement membrane, and they're just going to wind up going back into the blood. Occasionally they get stuck there, and that can cause an inflammatory reaction, and if we have time, we'll talk about that. The filtration slits just pretty much let everything through. All right, so what drives the filtration, of course, is hydrostatic pressure, and that is, in the case of blood, that's what blood pressure is. But remember that capsule is capturing this filtrate, and that creates a back pressure, so you have to look at the balance of those pressures along with a colloid osmotic pressure. Colloid osmotic pressure is due to the plasma proteins for the most part in the blood. So the glomerular hydrostatic pressure is the blood pressure in the glomerular capillaries, and it has to be high enough to offset the pressure in the capsule. So in glomerular capillaries, the pressure is higher than it is in peripheral capillaries. Typical peripheral capillaries have a blood pressure of maybe 35 millimeters mercury, but in glomerular capillaries, that pressure is up to 60. And part of the adaptation of that's a high pressure. Most capillaries would burst if they had a pressure as high as 60. So in effect, that's what the podocytes are doing. They're reinforcing the capillary walls and preventing them from bursting. The capsule has its own pressure. The capsular hydrostatic pressure is about 15 millimeters mercury. It offsets the blood pressure, and so that's why the glomerular capillary pressure has to be so high. But let me go back to that capsular hydrostatic pressure has to be high enough to push the fluid through the renal tubule the entire rest of the way. So it takes about 15 millimeters mercury of pressure in the capsule to force the fluid to flow through the proximal tubule into the nephron loop, through the distal tubule, and all the way through that system of collecting tubules. All right, so we have what we call the net hydrostatic pressure. So if the glomerular hydrostatic pressure is as high as 50 and the capsular pressure is 15, then that gives you a net of 35 millimeters mercury. In a typical capillary, say in connective tissue or muscle, a normal capillary pressure would be about 35, and a normal interstitial fluid pressure is about 0. So it works out to be about the same, but that capsular pressure has to be offset. The colloid osmotic pressure is due to the plasma proteins, and blood colloid osmotic pressure, no matter where you are in the body, is about the same. It's about 25 millimeters of mercury. So the net pressure of the blood is that, or the net pressure across the glomerular, the filtration membrane, is about 10 millimeters mercury. 35 was the difference between the blood pressure and the capsular pressure, and 25 is the colloid osmotic pressure of the blood. Remember, there are no plasma proteins in the filtrate, so it doesn't have an osmotic pressure. So that pressure of 10 millimeters mercury is enough to force filtrate to push through. And here's a diagram you need to stop and pay attention to. It's going to explain in more detail everything, pretty much everything I just told you. Okay, so the glomerular filtration rate is how much filtrate you produce, and we measure it each minute, but then you can magnify that to see how much a day. So 125 milliliters per minute, and that's for both kidneys together, and only about 10% of the fluid that's delivered to the kidneys actually leaves the bloodstream, but that still generates about 180 liters of filtrate a day, and that's about, your book says 70, I was thinking more like 60 times the total plasma volume. So we have to be able to regulate glomerular filtration rate. You know, sometimes you're overhydrated or you're dehydrated, so there's different reasons to lower the filtration rate or raise the filtration rate. And so the three ways we regulate that, and these are the three ways that we regulate blood pressure and blood volume body-wide. Autoregulation, which means something's going on at the local level, hormonal regulation, and autonomic regulation. So autoregulation of the glomerular filtration rate maintains the adequate GFR even if local blood pressure and blood flow change. And we can constrict or dilate the afferent arterioles, and even the glomerular capillaries. Typically, capillaries do not change their diameter. The arterioles control them. But remember, these capillaries have mesangial cells nearby that can help constrict them. Hormonal regulation is mostly through the renin-angiotensin-aldosterone system, but also through hormones that oppose those. These hormones are called natriuretic. Natriuretic means sodium in the urine. So we'll take a look at these as we go. The renin is secreted by the juxtaglomerular cells of the juxtaglomerular complex, and renin stimulates the activation of angiotensin, and locally that causes constriction of the blood vessels. Systemically, that causes systemic vasoconstriction, and it also can be stimulated by sympathetic innervation or by osmotic changes in the macula densa. The RAAS, that's a renin-angiotensin-aldosterone system, works by renin being an enzyme. It's dumped into the blood right there in the kidneys. So as blood is leaving the kidneys, imagine blood flowing through the renal veins, and renin is activating the inactive precursor angiotensinogen, which is produced by liver. It's just one of the plasma proteins that circulates all the time. But it converts it to an intermediate called angiotensin 1. Now, angiotensin 1 itself can't do anything, but it's just like a two-step conversion. So this is happening as the blood flows through to the inferior vena cava, then it's going to go to the right side of the heart, and from there it's going to go out to the lungs. And the lungs are lined with blood vessels that have this other little enzyme just kind of perched there, angiotensin converting enzyme, or ACE. So the ACE enzymes in the lungs are going to activate the conversion of angiotensin 1 to angiotensin 2, and that's the active player. Angiotensin 2 is a hormone, and it's circulating in the blood, and it stimulates the contraction of smooth muscle in the vessel walls, which causes vasoconstriction, and that's going to boost the blood pressure. It also stimulates the secretion of aldosterone by the adrenal glands, and aldosterone, we know, tells the kidneys to reabsorb more sodium, which generally prompts the reabsorption of water as well, so that increases blood volume, which also increases blood pressure. So the net effect of angiotensin 2 is to increase systemic arterial blood pressure. It also stimulates thirst center in the hypothalamus. The more you drink, the greater your blood volume. Increases production of antidiuretic hormone, which tells the kidneys to conserve water. So the overall effect of renin, angiotensin, aldosterone, and ADH is to increase systemic blood pressure and blood volume and bring you back to a normal GFR. So if we look back at what stimulated it, it was low blood pressure, or a change in the osmotic concentration, or sympathetic stimulation. So all of those are stressors. So the effect of RAAS is to increase blood pressure and blood volume. And again, here's a very complicated diagram in your book. You need to work your way through the diagram. Take your time. So hormonally, the GFR can also be regulated to increase the blood volume, and through RAAS mostly. The natriuretic peptides are going to do the opposite of that. There are two of these that we know of. Atrial natriuretic peptide is secreted by the right atrium, predominantly the right side, and it's secreted when you have a lot of blood. So the walls of the atria get stretched chronically because your blood volume is high, and that prompts them to secrete AMP. The ventricles are also known to secrete a parallel hormone called BNP, and my understanding is that the B stands for brain natriuretic peptide because it was first isolated in the brain. Anyway, the effect of ANP and BNP is to counteract the effect of angiotensin and aldosterone and ADH. So anything that we just talked about with the RAAS system, the natriuretic peptides are going to be antagonistic to that. So that's going to increase the loss of sodium, increase urine output, pull fluid out of the body, and then therefore lower the blood pressure. Autonomic regulation is mostly through those sympathetic nerve fibers to the kidneys, and mostly what that does is it causes constriction of not just the afferent arterioles, but the entire renal arterial system, renal arteries, and all of the arteries in between those and the afferent arterioles. Generally, you're going to do that if you need to shunt blood away from the kidneys to, say, your active skeletal muscles. But consequently, you're going to decrease the GFR and slow filtrate production. So you've got to have a counteracting intervention mechanism to keep this from completely shutting down the production of filtrate. Because if your kidneys aren't filtering any blood at all, then you're going to have a problem with rapid increase in metabolic wastes and homeostatic imbalance of electrolytes. So that sympathetic activation can be overridden by local mechanisms. Many of those are going to either cause constriction of the efferent arterioles or counteract and cause dilation of the afferent arterioles to keep filtration going, even if you've cut down blood flow to the kidneys. All right, filtration is passive. We're just passing the stuff through. Remember that in general, the proteins are not going to pass through, but it's not just metabolic waste in the filtrate. It's all of those nutrients, including glucose, amino acids, and other nutrients. So we've got to have a way to reabsorb the things we want to keep. And that's going to happen in the renal tubule, reabsorbing the useful organic nutrients, reabsorbing more than 90% of the water, and then additionally secreting any waste that we need to get rid of more than what we could just filter. So reabsorption generally is done in a two-step process. You're going to recover the nutrients through either active transport or secondary co-transport. You're going to have active transport of sodium ions and other ions, calcium ions as well. And then as you reabsorb all of those solutes, the reabsorption of water can occur passively through osmosis. Secretion is simply going to go in the opposite direction. You're going to have active transport of some things that you're going to secrete, and others may be passive, as we'll see. So reabsorption and secretion are the two processes that occur in the renal tubule, and to some degree in the collecting system. So we're going to use diffusion and osmosis and leak channels and carrier-mediated transport. If you need to go back to transport, then go back to the chapter on the cell and reread about membrane transport. We're going to use facilitated diffusion, meaning we're going to have some protein channels that are selective for particular things. Active transport, meaning we're going to use ATP. Co-transport and counter-transport. So carrier-mediated transport has some regulatory factors that are going to affect it. First of all, they're very specific. So a transporter that picks up glucose would not necessarily pick up a different sugar. They generally work in only one direction. So if you're reabsorbing them, they can pull them back out of the tubular fluid so that ultimately they wind up back in the blood. But if it's a carrier that is doing tubular secretion, it's going to put it into the filtrate. Different parts of the tube are going to have different specialties, different things they pick up, and different parts of the cell even can have different kinds of carriers. So there's an aspect of how we measure the transport of these things. It's called the transport maximum. And in general, what I want you to remember that the transport maximum is a measure of how much you can absorb. If it's a nutrient that you want all of, it's going to have a high transport maximum. If it's a waste product that you're mostly trying to lose, then it's going to have a really low transport maximum. So transport maximum is simply a measure of how well you can reabsorb. As an example, the transport maximum for glucose is about 320. If you get blood glucose that's too high, it exceeds that renal threshold, and then you can't reabsorb it all. The units your book uses are different, 180 milligrams per deciliter, and any higher than that, and the glucose kind of spills over into the urine. A condition called glucoseuria, which is associated with diabetes mellitus, but it doesn't have to just be that. So a normal plasma glucose, remember, is about 70 to 110 milligrams per deciliter, so anything higher than about 180, and glucose can appear in the urine. It could just be that you ate a lot of sugar. If you sat down and ate a huge whopping helping of sugar, a box of cookies, and washed it down with Kool-Aid, then you could exceed that renal threshold for glucose, and your urine will smell sweet. For amino acids, the renal threshold is about 65. You don't need to remember that. But if you remember from metabolism, you don't store proteins. So proteins that are picked up in the liver are deaminated and burned, but if you get your blood amino acid level high enough, yeah, you're going to lose amino acids in the urine. And that doesn't mean there's something wrong with you or your kidneys. All right, osmotic concentration, body fluids, just to have a benchmark, have a concentration of about 300, and that includes the blood, 300 milliosmoles per liter. Ions we're going to report in milliequivalents per liter, and organic molecules we're reporting in milligrams per deciliter. Okay, just so you have some idea of the units. So as we look at this table, again, we're comparing reabsorbed to secreted. We reabsorb the ions sodium, chloride, potassium, calcium, magnesium, sulfate, and bicarbonate. They're mostly reabsorbed, but look at what we secrete. Potassium, hydrogen, calcium, and phosphate. So you see both hydrogen and calcium on both lists, and it just depends on how much you ate. So if you ate a lot of calcium, if you ate more calcium than you need, then yeah, you're going to secrete it. Metabolites, glucose, amino acids, proteins, and vitamins are all reabsorbed. Waste products, creatinine, ammonia, and the metabolic acids and bases like urea and uric acid are going to be secreted. And then there's some miscellaneous things which we're just going to make note of. If you have an excess of neurotransmitters in your bloodstream, then you're going to excrete them. Histamine, you're going to excrete. Drugs like penicillin, you're going to excrete. And then there's no transport mechanism for urea, water, urobilinogen, or bilirubin. Urea and water can be reabsorbed passively. Urobilinogen and bilirubin are too big to reabsorb. So once they are lost in the filtrate, they're just not going to be reabsorbed. Reabsorption and secretion at the period or proximal convoluted tubule, about 60 to 70% of the total filtrate volume, and 100% of the organic nutrients, and most of the ions, most of the water, and a little bit of secretion there. So sodium ion reabsorption here is mostly simply by either diffusion through leak channels or co-transport of organic solutes. So sodium co-transport is an important process. And sodium can also be counter-transport for hydrogen ions, and later on we'll see for potassium ions as well. So looking at this diagram, look at the key. Water reabsorption, solute reabsorption is very high in the proximal tubule. Secretion is variable, and we'll look at that separately. I love this diagram because I just like learning about this stuff, but it is kind of busy. Look at your key to make sure you understand which things are being secreted, that's red arrows, and which things are being reabsorbed, that's the blue arrows. Okay, reabsorption and secretion along the nephron loop. We've already got 60 to 70% of it reabsorbed before the fluid even hits the nephron loop. So we're down to 30-40% of our total 180 liters a day. And we're going to get about half of the remaining water and about 2/3 of the remaining salts. And we're going to do that in a very weird way. In the descending limb, we can let water in or out, but solutes cannot easily leave. The descending thin limb is going to reabsorb sodium and chloride ions from the tubular fluid. The ascending limb, though, is impermeable to water, and it is very permeable to sodium and chloride, but only in one direction. And we're going to see that's even more exaggerated in the juxtamedullary nephrons. So reabsorption and secretion at the distal convoluted tubule, we're down to maybe 15-20% of the total volume we started with. Pretty much all that's left is electrolytes and metabolic wastes and some fluid. And a lot of those metabolic wastes, remember, we're just trying to get rid of them. And the electrolytes we're going to be able to absorb if we want them, and not if we don't, because a lot of those are under hormonal control. So we can actively secrete ions we want to get rid of, acids that we need to dump, certain drugs and toxins, which we're not going to look at the mechanisms for those. Selectively reabsorb sodium and calcium. Selective means under the control of our hormones. Remember, our hormones are aldosterone for sodium and calcitriol and PTH for calcium ions. And then the amount of water we absorb, we'll see, is also controlled. At the distal convoluted tubule, we're going to actively transport the sodium and chloride out of the tubular fluid. That's going to make it more dilute. And then in the distal regions, we're going to have pumping of ions as an exchange. Sodium usually for potassium, as we'll see. So here is a small diagram showing you in the distal convoluted tubule, chloride and sodium being reabsorbed into the blood, sodium being exchanged for potassium. So in general, you get rid of potassium and you keep sodium chloride. Aldosterone is the hormone that's going to control that process. Aldosterone is produced by the adrenal cortex. Remember, it was stimulated by angiotensin. It can also be stimulated by high levels of potassium in the blood. Potassium, remember, is supposed to be way low, 3.5 to 5 milliequivalents per liter. And you have to regulate potassium very carefully, because if you don't, it can affect neuron excitability and affect the heart. So potassium is very strongly regulated by aldosterone. So aldosterone dumps potassium and reabsorbs sodium in its place. So here you can see the aldosterone-sensitive process of potassium and sodium exchange. And this isn't in all of the cells of the distal convoluted tubule, it's just in specific cells that are aldosterone-sensitive. All right, so what happens if you don't control potassium? You can have hypo or hyperkalemia. Hypokalemia means obviously it's too low. In general, you're not going to have high potassium through dietary ingestion. Actually, it's hard to mess with your potassium. If you don't have an aldosterone problem, you probably don't have a potassium problem. But one thing you ought to keep in mind is that potassium chloride is one of the components of death by lethal injection. They use potassium chloride and give hyperkalemia to shut down nerve impulses. So whether it's hyper or hypokalemia, it's bad, and we'll study that in more detail when we get into electrolyte balance in the next chapter. We already saw that atrial natriuretic peptide opposes aldosterone, so it's going to have the opposite effects. And then parathyroid hormone is going to regulate calcium reabsorption, and it's going to increase the reabsorption of calcium at the distal convoluted tubule. So secretion at the distal convoluted tubule is mostly for things like potassium and hydrogen ions that you're trying to regulate. And potassium ion secretion is controlled by aldosterone. And hydrogen ion secretion we'll look at in more detail next chapter. Hydrogen ion secretion is going to be associated with pH regulation. Remember, hydrogen ions are associated with high acid or low pH. And bicarbonate ions go along with that. So we'll look at that in more detail in the next chapter. All right, so here's just a diagram showing you hydrogen ion secretion. I think we'll defer that until we get to acid-base balance in the next chapter. Control of blood pH then is going to be an important function of the distal tubule. Proximal and distal tubule both have cells that can deaminate amino acids. Remember, this means pulling an amino group off, but they're going to just dump it right into the urine. It's not going to get into the blood, so there's no need to convert it to urea. It can stay in the form of ammonia and be buffered into ammonium ions. And that, again, is going to be associated with acid-base balance. So we'll come back and talk about that. If the kidney cells are reabsorbing amino acids from the filtrate, they may just be burning them. They deaminate them and burn them in the Krebs cycle. So they're just using that for ATP production or for the needs, if they need amino acids of their own. All right, as we get into the collecting system, the main component is going to be the collecting ducts. And these, we find, have cells that are hormone sensitive. Aldosterone controls some of them, and ADH controls others. So this idea of exchanging sodium for potassium that we saw in the distal convoluted tubule also applies to the cortical region of the collecting ducts, and the acid-base balance functions of the distal tubule also apply to the cortical collecting ducts. Urea is kind of funny. Urea is, it's technically lipid soluble, but it's really small, and it can get through plasma membranes without a whole lot of difficulty, so you don't need a transporter for it. It can get reabsorbed by diffusion. Secretion in the collecting ducts, likewise, is for pH control, so we'll come back and talk about that later. As we get into this concept, this one's really kind of tough. The countercurrent multiplication is a system that uses the juxtamedullary nephrons with those long loops of Henle and concentrates the solutes in the renal pyramid. The basic story is that this countercurrent mechanism holds salts and urea in the pyramids, and that gives an osmotic effect which helps draw water out of the collecting ducts. So you've got the two opposing limbs, the descending and the ascending limbs, and they are very different in terms of their permeability. The descending limb is permeable to water but not solute, and the ascending limb is permeable to solute, sodium and chloride particularly, and not water. And so basically what you can do is pump salt out of the thick ascending limb. That's right before it goes back up to the distal convoluted tubule. And that means the tubular fluid is more dilute, but that salt stays in the pyramid. And it draws water out of the descending limb. And that water then can be pulled out through those vasa recta, those capillaries associated with these long loops. So we're going to get concentrated solution arriving in the thick ascending limb because we pulled the water out in the descending limb, and that makes it easier to pump even more salt out into the peritubular fluid so that by the time the fluid leaves the loop, it is a whole lot less in total volume and it's more dilute than it was. So that gives us this osmotic gradient in the renal pyramids, which are in the medulla, and it keeps a lot of salt in the pyramids. We're not that concerned about the specific mechanism of how many sodiums, potassiums, or chloride, but basically we're using a co-transporter to take that salt out of our tubular fluid and put it in the medulla. And so when you look at the diagram on the left, just showing the loop, you can see where water is leaving in the descending limb and salt is leaving in the ascending limb. And then you don't have to worry about the specific mechanism shown up close on the right. But here you can see the effect of that. So we're looking at the renal medulla, and we're looking specifically at the loop, and we're seeing the change in osmotic concentration. 300 is normal for body fluids, but as more and more water leaves the descending limb, that fluid becomes more and more concentrated, up to 1200. And then as it goes back up the ascending limb, we pump the salt back out. The water's already left. So we pump the salt back out and we get it actually more dilute, only about 100 as it goes into the distal convoluted tubule. But the second part of this is that salt that we just pumped out, a lot of it is going to stay put. And the reason it's going to stay put is because this is a countercurrent mechanism. Okay, so we have salt being pumped out right before it moves into the distal convoluted tubule, so it actually has a very low osmotic concentration of only about 100 milliosmoles per liter. Okay, and again, the whole point of that is to create the osmotic gradient in the medulla, and the medullary osmotic gradient is about 100 to 300 at the top, down to about 1200 milliosmoles down at the papilla of the pyramid at the very base. And a lot of that's the salt, but urea also, because it's little lipid-soluble molecules and it's highly concentrated in the filtrate. By now you've already removed 80% of the water and you've got all of that urea left, so it can diffuse out, but it's going to stay in those pyramids. So between the urea and the salt, they're going to create a strong osmotic gradient that's going to pull water out of the collecting duct. So here you can see from the end of the proximal tubule at the top left, a milliosmolarity of 300, down to about 1200 at the flexure point of the loop, up to about 100 in the distal convoluted tubule. And then what happens is that the papillary duct, which is permeable to urea, lets urea leak out. That's the little orange thing. And the upper portions of the collecting duct can be permeable to water. It depends on the presence of the hormone, antidiuretic hormone. So we're going to regulate urine volume and concentration through controlling water reabsorption, and we're going to control water reabsorption in the loop of Henle variably using the hormone ADH. So in the proximal and distal tubule, we're just going to reabsorb almost all the water. The only place we can control it is in the collecting system. So by the time fluid gets to the distal convoluted tubule, you only have 1 to 2% of the water volume left. Out of that 180 liters a day, that's all you have left. And all of that was obligatory water reabsorption. You can't stop it. Up to 85% of it is obligatory. And then the part you can control is called facultative, and that's under the influence of antidiuretic hormone. And the way antidiuretic hormone works is that it opens aquaporins, which are water channels, in the collecting duct cells, and that lets the water leave the tubular fluid and get into those really salty pyramids. So with ADH, you reabsorb a lot of water. Without ADH, you're going to not reabsorb that water. You're going to have higher urine output. ADH is produced in the hypothalamus, secreted by the posterior pituitary. Secretion of ADH is stimulated by being dehydrated. So if the osmotic concentration of your blood gets a little too high, higher than 300, it stimulates nerve impulses from the hypothalamus to the posterior pituitary so that you secrete ADH. ADH circulates to the kidneys. It opens those water channels in the collecting ducts, lets water leave the tubular fluid. So one way to remember that, let's go back to that slide. ADH secretion is problematic in children that are learning bladder control at night. So if you have bedwetters, usually giving them a little ADH at night actually helps them sleep through the night. Old people tend to secrete less ADH, so they have to get up and go to the bathroom at night. People who can't secrete ADH may have a condition called diabetes insipidus, when the water just passes right through them. They're always thirsty, they're always drinking, and they're always peeing because they can't control their water output anymore. And alcohol, excess alcohol consumption can inhibit the secretion of ADH. So people that drink too much alcohol very often wake up with a very dry mouth because they're dehydrated, and a very full bladder because they didn't secrete enough ADH. And so they got dehydrated on top of too much alcohol effects on the brain. So the osmotic concentration arriving at the DCT is 100. If ADH is not secreted, it stays that way, but in the presence of ADH, the osmolarity can approach that maximum that we saw in the pyramids, which was 1200. So that's 12 times as much of a concentration. So this diagram shows you where the water's leaving in the descending limb, where the sodium and chloride are being transported out in the ascending limb. And in this case, antidiuretic hormone is not being secreted. And in the lower figure, you see that it is. So ADH is being secreted and water is leaving in large amounts. So you're going to have a small volume of concentrated urine in the presence of ADH. In the absence of ADH, in the top picture, a large volume of dilute urine. Okay, so the hypothalamus is secreting this ADH. And, you're going to have a threshold low level that's always going to be present. But you can increase or decrease that depending on the state of hydration. So if you drank a bunch of water, you're going to decrease your ADH secretion and produce more urine. And if you aren't drinking enough water, then you'll secrete more ADH and conserve more water and produce less urine. So a normal amount of urine produced every day is somewhere around 1 to 2 liters. And again, it depends on how much water you drink. So 1 to 2 liters is considered normal. All right, we didn't talk about the role of the vasa recta, so let's go ahead and get that out of the way. Let's take a look at a picture. Oh, I don't have a picture, sorry. So the vasa recta are going to parallel the nephron loop, and basically their function is to carry off the extra water and salt that you reabsorb without ruining that gradient. Okay, so we've got a summary here. I think I'm just going to leave you to read through that. I don't need to read that to you. But basically this is just pulling all of the steps together. And we end at the papillary duct, and that is where we're going to pass into the minor calyx. And again, humans cannot alter urine concentration or composition or volume after that point. Okay, so be sure you read through that. Composition of urine, of course, depends on how much you ate and other things, but a typical composition of urine is going to have, it's mostly dilute, even though you can make it more concentrated. It's sterile until it gets to at least to the urethra. It's supposed to be sterile. The color of it, the yellow color, is due to the pigment urobilin. And urobilin is actually a derivative of the bilirubin that was a breakdown product of hemoglobin. Some of that bilirubin that you secrete in bile does get reabsorbed, and if it gets into the kidneys, it gets converted to urobilin, which is sometimes called urochrome, the pigment of the urine. And it comes from urobilinogens, which are derived from the bilirubin in the intestine. All right, so here are the normal values. I want you to pay attention to normal pH. The average is 6, but the range is 4.5 to 8. So from very acidic to pretty darn basic. And compare that to the normal pH of blood, which is 7.4. So a pH of 6 being average means that most of the time you're secreting more acids than you're keeping. Specific gravity is a measure of how concentrated the solutes are. The specific gravity of pure water is 1. So 1.003 is barely any solute. It's almost completely water. But the high end of the range for humans, 1.030, is only 10 times as much, and that's still not a lot of solute. So our urine is mostly water no matter what we do with it. Osmotic concentration, water content, volume, color, clarity, odor, bacterial content, of course, should be none. All right, so there's some mechanisms we use to test kidney function, and one of these is creatinine clearance, and another is blood urea nitrogen. Creatinine clearance is simply a measure of how much is in the urine with how much is in the blood. And the thing about creatinine is you don't reabsorb any of it. So any creatinine that was filtered at the glomeruli is going to wind up in the urine. But a problem with using creatinine is you do actually actively secrete by tubular secretion some creatinine. So to measure that as a measure just of filtration doesn't exactly work. We use another compound called inulin. Please don't get that confused with insulin. Inulin is something they can actually inject into you. They can measure the inulin concentration in your blood, compare it to what's in the urine, and that will give them a measure of glomerular filtration rate. That's beyond what I'm going to expect you to remember for the test. Blood urea nitrogen, you'll see this sometimes on urinalysis reports, the BUN report, and it measures the amount of urea in the blood, and it's supposed to be low because urea is a waste product. So we're going to modify the filtrate. We're going to talk about urine production, but remember it's all going to end at the renal pelvis, and from there we've got to get the urine transported, stored, and eliminated. So the ureters are the tubes on the right and left that connect the kidneys down to the urinary bladder. They're not passive. They have muscle in them, and they work by peristalsis to propel urine down. They are retroperitoneal. Recall that this means that they are behind the parietal peritoneal wall, but then the kidneys are retroperitoneal. They're not completely retroperitoneal. There's a point at which they have to penetrate through the peritoneal wall, and that's a point where it can get occluded or kinked and cause backflow of urine. The orifices, the ureteric orifices, are just the openings into the bladder, but they are pretty much pinched shut to help prevent backflow unless urine is actively being moved by peristalsis. So you've got, it's a mucous membrane, so you have a mucosa with a transitional epithelium, muscular layer to give you peristalsis, and then the outer layer is continuous with the peritoneum. Peristalsis occurs, it says here, about every 30 seconds. You can't feel it. It's just going to be as urine accumulates in the renal pelvis, it stretches it, and that triggers peristalsis. So that propels it all the way into the urinary bladder. Urinary bladder can contain, it says here, up to 1 liter of urine. I'm sure that depends on how old you are, whether you're pregnant or not, and other factors. Some people just have a better urinary retention than others. This is a male, and we can tell this is a diagram of a male because it has a prostate gland surrounding the urethra. Enlargement of prostate, particularly in older males, is a problem in that it restricts urine outflow, so it takes longer to empty the bladder. Notice the interior of the bladder has rugae. That's similar to the stomach. That allows it to stretch. The detrusor is the bundles of smooth muscle, and they're bundled so that, again, the bladder wall can stretch out. A fully expanded urinary bladder is so thin that the wall looks practically translucent. There are two sphincters. The internal urethral sphincter at the outflow of the urinary bladder is smooth muscle. The external urethral sphincter, which is in the floor of the pelvic cavity, the pelvic floor, is skeletal muscle. So the mucosa of the urinary bladder is folded in a rugae. There's a smooth area, I didn't mention that, the trigone, which is the connection of the three orifices, and that part doesn't expand. So it kind of anchors the urinary bladder and funnels the urine flow out through the urethral orifice. The neck of the urinary bladder is the outflow region. It has the internal urethral sphincter. The innervation of the urinary bladder, you've got sensory fibers which respond to the stretching of the bladder, and then you have parasympathetic fibers to control contraction of the bladder, and these are by pelvic nerves from the hypogastric plexus. The wall of the bladder has the layers mucosa, submucosa, and muscular layer. The detrusor muscles, remember, are bundles of muscle which kind of separate out as the bladder expands. The urethra transports urine from the urinary bladder to the exterior. It has, again, folds in it to allow expansion. It's transitional epithelium. The external sphincter in both sexes is skeletal muscle. It is under learned voluntary control through a nerve that supplies the pelvic organs called the pudendal nerve. And it has resting muscle tone. All that means is it's mostly closed. It only opens when you tell it to. It's voluntary relaxation. And of course that has to be learned. So the reflex is associated with urinary storage reflex. Basically inhibits contraction of the detrusor muscle so that the bladder will expand. And then you learn, the pontine center is in the pons. You remember the pons is in the brainstem. So that's a center where you learn to inhibit urination. And there you can see the diagram. Now I want to follow this through because part of it is visceral, part of it's parasympathetic, and part is somatic and under voluntary control. So let's start with the stretch receptors in the bladder, and they go to the sacral region of the spinal cord, and notice there's a reflex up to the lower thoracic and upper lumbar spinal cord to go out to an autonomic ganglion to inhibit contraction of the detrusor muscle, and this helps to stop urination. So this is the way you're going to stop it. And then you also have a local reflex to the internal sphincter, and then from the pons you're going to go from the brainstem down through the spinal cord, again to stimulate the external urethral sphincter and prevent the bladder from emptying. Okay, so to empty the bladder, to void urine, involves some different spinal reflexes, and you're going to see here that the stretching of those receptors in the bladder is going to stop the inhibitory response and stimulate a parasympathetic pathway that tells the detrusor muscles to contract. But look up at the pons. We're also getting the urge along the red pathway, the afferent pathway. We're getting the urge to urinate because those stretch receptors are activating that conscious sensation. And in this case, the pontine center is going to stimulate the opening of the external urethral sphincter. That's an inhibitory thing. Now that sounds backwards, but think it through. Skeletal muscle contracts when you tell it to. So if you're not telling it to contract, it's going to relax. So the only part of this muscle that's being activated to contract is that parasympathetic pathway in the sacral region that's telling the detrusor muscles to contract. So in infants, of course, there's no voluntary control because, first of all, they haven't learned it, and second of all, the nerve pathways are not even there. So incontinence occurs when you cannot control urination voluntarily, and it may be trauma to the sphincters, or it may be loss of, let's look back at the picture, loss of the pathway. If you have transection of the spinal cord between the pontine center and anywhere down to those lumbosacral areas, then your pons cannot tell your detrusor muscle to contract. It cannot tell your external sphincter to relax either. But if you have damage directly to that sacral region of the spinal cord, you lose the ability to tell the detrusor muscle to contract. So damage above that region gives you incontinence, but damage at that region prevents the bladder from emptying. And in some cases, the bladder can expand to the point of bursting. More likely, it's just going to leak continually. Okay, so be sure that you go through the interactions with other body systems. Review this presentation if you find it helpful, but remember that your test questions are going to come from the posted review materials in Canvas.